Have you ever taken a drink at the wrong moment, laughed, and had it all spray out of your nose? It happens to the best of us, but it's pretty uncomfortable and embarrassing. Luckily, it doesn't happen all the time, and we have velopharyngeal closure to thank for that. The uvula, or soft palate, is a comical-looking structure that we can see at the back of our mouth when we look in the mirror. It resembles a boxing ball that Jerry the mouse would often punch when eaten by Tom the cat in those classic cartoons. It plays an important role in speech to produce nasal sounds, but it is also essential in the oral transit stage of swallowing to make sure the bolus stays well away from our nostrils. In this session, you're going to learn why velopharyngeal closure occurs, the anatomical structure of the uvula, the mechanism of velopharyngeal closure, how the brain controls velopharyngeal closure, and which cranial nerves innervate the uvula. Before you start, make sure you print out the velopharyngeal closure worksheet to make notes as you progress through this video. Okay, let's get started. So by now, you will have picked up on the importance of the uvula making a tight seal against the upper pharyngeal walls to block food or drink from entering the nasal cavity and causing us some discomfort and embarrassment. Velopharyngeal closure also prevents air escaping through the nose when we swallow, which allows a degree of pressure to build up and be maintained in the oral cavity and pharynx when we swallow. Let's look at the anatomy of the uvula so we can understand just how something so floppy lifts up and down. The main body of the uvula is made up of a muscle called the musculus uvulae. One half of the muscle anchors itself to the palatine aponeurosis, a layer of fibrous tissue at the roof of the mouth. The other half of the muscle attaches to the mucosa surrounding the surface of the uvula. When the muscle fibers contract and shorten, they cause the uvula to become short and stubby in shape, but they don't cause it to lift up. That's the job of some other muscles. There are two other muscles involved in velopharyngeal closure. They're a bit trickier to see, so we have to turn the head around and look at it from the back. The first of these muscles is called the levator veli palatini. One half of the muscle anchors itself to the palatine and sphenoid bones on the base of the skull. The other half of the muscle attaches to the palatine aponeurosis, the same spot as the musculus uvulae. When this muscle contracts and shortens, it elevates the palate. It's easy to remember. Levator, levitate, lift up, get it? The other muscle is called the tensor veli palatini. One half of the muscle attaches to the sphenoid bone only, while the other half attaches once again to the palatine aponeurosis. When this muscle contracts and shortens, it makes the floppy uvula tense and firm. Again, it's easy to remember. Tensor, tense, firm, beginning to make sense? So by understanding the anatomy of the uvula, we can now begin to understand what is needed to achieve effective velopharyngeal closure. The uvula needs to become short and stubby. It needs to become tense and firm. And then it needs to lift up, making firm contact and a tight seal with the walls of the nasopharynx in the process. That's a lot of information to take in, so take a moment to fill out your handout with what you've learnt so far. Okay, let's keep going. We're going to help you understand how the brain gets the uvula to move in the first place. Unfortunately, velopharyngeal closure is innervated by two different cranial nerves, so it's going to take a bit of explanation. Let's start with the tensor veli palatini muscle. A signal is generated in the primary motor cortex on the surface of the brain and travels down the length of the neuron cell through the corticobulbar tract. Remember, these neurons in the brain sending signals to move muscles are upper motor neurons. When the end of the neuron reaches the part of the brainstem called the pons, it will transfer the electrical signal from the upper motor neuron to the lower motor neuron in a region called the trigeminal nuclei. This is called synapsing. From there, all those lower motor neurons together will make up the trigeminal nerve, which is cranial nerve number 5. The trigeminal nerve is a thick nerve that emerges around the middle of the pons. It will exit the base of the skull via a hole called the foramen ovale. The electrical signal will then travel along the trigeminal nerve, specifically down the mandibular branch. When the signal reaches the tensor veli palatini muscle, the uvula will become firm. When it comes to the musculus uvuli and the levator veli palatini, the initial electrical signal in the primary motor cortex is generated and travels down the corticobulbar tract. However, this time the upper motor neuron travels to an area on the medulla called the nucleus ambiguus. This is where the upper motor neuron synapses with the lower motor neuron, which forms the vagus nerve, cranial nerve number 10 this time around. It emerges from the sides of the medulla. It travels out of the skull via the jugular foramen towards the musculus uvuli and the levator veli palatini. So both the vagus nerve and the trigeminal nerve are involved in achieving velopharyngeal closure. But sensory innervation is also important in this process. It ensures that you can feel if you have achieved full closure. Unfortunately, if anything has escaped up your nose, it also gives you that unpleasant feeling. The mucosa on the posterior or back of the uvula, as well as the walls of the nasopharynx, are innervated by a different cranial nerve called the glossopharyngeal nerve, cranial nerve number 9. If any food or drink escapes upwards towards your nasal cavity when you swallow poorly, the glossopharyngeal nerve will send a signal up to the brain. The nerve enters the skull via a hole called the jugular foramen and merges with the brainstem in the medulla. The glossopharyngeal nerve carries information about general sensation, which is touch, temperature, and pain, just like the trigeminal nerve does for the face and oral cavity. Therefore, both nerves, the trigeminal and the glossopharyngeal, share one same nuclei in the brainstem, which is the spinal nuclei found within the medulla. The neurons of the glossopharyngeal nerve will carry the electric signal to the spinal nuclei in the medulla and transfer it to the neurons in the brain in a process called synapsing. The electrical signal will travel up the neuron axon in the brain via the thalamus. The thalamus is a special area deep within the brain that is important for processing sensory information. From there, the electrical signal continues its journey up the neuron until it reaches its final destination of the primary sensory cortex. Once this happens, you realize that you have felt your palate lift up against your upper pharyngeal walls successfully to prevent nasal regurgitation. If something went wrong, however, you'll probably feel something very uncomfortable up the back of your nose as well. That's a lot of information to take in, so fill in the rest of your handout. When finished, it should look something like this. Fantastic work! In the next session, we're going to look at how the tongue pushes the bolus backwards in a process called oral bolus propulsion.